everyone. I am Shurashree Pal. I am a Prime Minister's Research Fellow from IIT Kharagpur. And today we will be solving a GATE 2023 question from the paper Life Sciences, question number 43. Now, this is a multiple select question, so which means that there will be more than one correct answers. So the question states, B cells produce two forms of immunoglobulin, membrane-bound form known as B cell receptor and its soluble form known as antibody. Which of the following statements is or are correct about BCR and antibody produced by the same B cell? The options are A. BCR and antibody have identical antigen binding site. B. BCR and antibody recognize different epitopes. C. BCR and antibody are encoded by the same gene. And D. BCR and antibody are formed by differential splicing. So, for more information on the structure of antibody, B cell receptor, you can follow this immunology course by NPTEL, and the YouTube link is also provided here. So, we will discuss a little bit about the immunoglobulin, its structure, and what is the difference between a B cell receptor and the soluble form of the immunoglobulin, which is the antibody. Now we know the B cell antigen receptor or the B cell receptor, it is actually formed from the same genes that encode the antibody. So the B cell receptor and the antibodies, they are coded by the same gene and they form this class of proteins known as the immunoglobulins. The antigen receptor of the B cells, they are known as the membrane immunoglobulin because they are bound to the membrane. So the receptor, the cell surface receptor on the B cell, it is attached to the membrane. So it has a transmembrane part of this protein, so which helps it to attach to the membrane, and that is why called the membrane immunoglobulin. Whereas, or you can also call it surface immunoglobulin. Whereas the antibody molecule, which is secreted out of the B cells, since it is secreted, it does not have a transmembrane region, so it is secreted out of the B cells, and those molecules are called antibodies. Now, when antigen binds to a B cell antigen receptor or a B cell receptor, or also called the BCR, the B cell proliferates and differentiates into plasma cells. Now, these plasma cells, they are the effector B cells. Now, effector B cells means that they secrete antibodies and have the same antigen specificity as the plasma cells B cell receptor. So here we need to understand this very clearly, that how do the B cells secrete antibody? So when the B cells, when they the receptor on the B cell, which is the B cell receptor, when they bind to the antigen, so that generates a signal in the B cell, and because of this signal, the B cell starts to differentiate, starts to proliferate, and these differentiated B cells now become the effector plasma cells. Now, the difference between a normal B cell and a plasma cell is that the plasma cell can secrete these B cell receptors as the antibody molecules, right? So these plasma cells B cell receptor and the antibodies that it secretes, both of them have the same specificity in terms of antigen binding. So what does it mean? So we know that this is the structure of an antibody molecule. So in the antibody molecule, there are typically two chains, two light chains and two heavy chains. So these two, the yellow ones, these are the light chains, and these two are the two heavy chains. Also, there are two regions of any antibody molecule. That is, one is the constant region, or which is also called the FC region, or the fragment crystallizable. The FC stands for the fragment crystallizable, and the FAB region. FAB stands for fragment for antigen binding, and this is the antigen binding site or the variable region of the antibody. Now, this through this variable region, in the variable region, there are these complementarity determining regions or CDRs through which will actually be specific for the epitope that it wants to bind to. So this is specific to the antigen. So that gives the specificity of the adaptive immune response, specificity of the antibodies against the antigen comes from these CDRs. So this is the antigen binding or the variable region of the antibody, and this is the constant region. This part is the constant region, which is actually responsible for the effector functions of the antibody. Now, um, this is the structure of the molecule which is secreted out of the plasma cells. And through these antigen binding site, this antigen binding site of the antibody is actually identical to the antigen binding site of the B cell receptor. So they have same specificity towards the antigen. So, which is why the antigen that activates a given B cell becomes the target of the antibodies produced by the B cell's progeny. The same antigen which actually activates the B cell that can be now targeted by the antibodies which will be produced by the same B cells progeny, which will be the plasma cells that secrete the antibodies against the specific antigen. Now, to give you a bit more detail about it, so we know I was talking about these epitopes and antigen binding, and this is, I've already told you that this is the antigen binding site of the antibodies, and this yellow molecule, this is an antigen, and these antigens can have different epitopes, can have multiple epitopes, and the antibodies are specific to these epitopes. So this particular blue antibody, we can see that it binds to this particular um, epitope on the antigen. Okay, so this molecule of antibody is specific to this epitope, whereas this red molecule of antibody is specific to this particular epitope on the antigen. So two epitopes of the antigen are shown here, one in the blue and another in the red. So here, this particular antibody is specific to this epitope, and this antibody is specific to this epitope. So therefore, depending on the epitope of the antigen, the antibodies is specific to them, specific to the epitopes, and that is why the antigen-antibody interaction is called very, very specific. Now, the membrane-bound B cell receptor, it does not have any effector functions. So I already mentioned that the effector function of the antibodies, that means the way it helps to neutralize the antigen or any pathogen, or it provides the immune response in the host. Uh, these functions, these are the effector functions of the antibody. And the membrane-bound B cell receptor does not have these effector functions. Why? Because the C region remains inserted in the membrane of the B cell. C region means the C terminal part of this particular protein. It remains inserted in the membrane of the cell. So it is a transmembrane region. And the function of the B cell receptor is to recognize and bind antigen via the variable regions exposed on the surface of the cell. So for the BCR, as you can see here, this is a B cell receptor complex. So the BCRs, these antigen binding portions are on the surface of the cell, and through these antigen binding sites, they can bind to the antigen, and then it transmits a signal that activates the B cell. Now, leading to clonal expansion and antibody production. So that particular B cell, which is activated by the antigen, will be clonally expanded. So it will produce its clones so that it can produce its antibodies. Right now, this is the B cell receptor complex. So the B cell receptor alone is not enough to produce the signals when it binds to the antigen. So it needs two other molecules, Ig alpha and Ig beta. These are the signaling molecules that actually form a complex with the B cell receptor, and together they form the B cell receptor complex. So these are the signaling molecules, the Ig alpha, Ig beta. So once the antigen binds to the B cell receptor, these signaling molecules help the signal of antigen binding to transmit into the cell through these ITAM motifs, so which are immunotyrosine associated motifs, which help these molecules to transmit the signal of antigen binding to the cells now, so that now the cell can proliferate and differentiate. Now, the structure of the B cell receptor, it is identical to its corresponding antibody, except for a very small portion at the C terminus or the heavy chain constant region. So, as I mentioned, that the C terminal region or the heavy chain constant region um, of this B cell receptor is actually transmembrane. So in the B cell receptor, the carboxy terminus is a hydrophobic amino acid sequence. So since it is a hydrophobic amino acid sequence, it will actually go inside the hydrophobic cell membrane. So it helps to anchor the molecule in the membrane. And the antibody, this is a hydrophilic sequence. So the carboxy terminus of the heavy chain, it is a hydrophilic sequence in the antibody molecule, which allows the antibody to be secreted and not bound to the membrane. Okay, so the hydrophobic amino acid sequence comes in the B cell receptor, which has to go into the cell membrane, and in case of the antibody, there comes the hydrophilic sequence, which is not attached to the receptor, not attached to the surface of the cell anymore. It is secreted. Now, how come the B cell receptor and the antibody, which is the secreted form, so how come they gain these different amino acid sequences? One hydrophobic, another is hydrophilic. And when they are coded by the same genes. So as I mentioned in the very beginning, that these two molecules, the BCR and the immunoglobulin, which is secreted, both of them are coded by the same gene. So how do they gain different amino acid sequences at their C terminus? So that is explained by differential splicing, or which we also call alternative splicing. So this is actually the phenomena that helps to generate the two forms of the antibodies. One is transmembrane, another is secreted. So this we can see in the topmost line, that this is the DNA. Actually, this is the rearranged DNA, because we can see the VDJ sequence has already been rearranged here. And these are the constant regions of this immunoglobulin molecule. See the exons corresponding to the constant regions, CMU1, CMU2, CMU3, CMU4, and so on. Now here we also should carefully look at this, that there are some other sequences like this one, PAS, which is actually the polyadenylation sequence here, and this is the PAM, which is another polyadenylation sequence. So why are these two polyadenylation sequences present here? We will see. There are also these two regions here, M1 and M2. Now, coming to that, now we have, first of all, from this DNA, the primary mRNA transcript will form, and then it will be processed. This particular RNA will be now processed. And as we know that the RNA processing involves splicing, which is the removal of the introns and the joining of the exons. So these intron parts, these small parts, the intron parts, they are spliced out and these exons are joined. And in case of the transmembrane IgM, these two sequences, the M1 and M2, they are also joined at the C terminus of, or sorry, not the C terminus, at the 3' end of this particular mRNA. And for this transmembrane IgM or the membrane-bound IgM, the polyadenylation sequence, this PAM is active. So now here the polyadenylation is carried out at this end. Okay, whereas what happens in case of secreted form of the IgM, this particular these two sequences M1 and M2, they are spliced out. So that is alternative splicing. So for the same gene, the splicing is somewhat different in these two. So here we can see that this particular polyadenylation sequence is recognized, and therefore the polyadenylation is done at this end. And hence the M1 and M2, these two sequences are spliced out from the premature mRNA. Therefore, these two sequences M1 and M2, they are spliced out in the secreted form, and here these two sequences are not present in the mature mRNA. Now, what is the benefit of these two sequences? These two sequences actually help to form the carboxy terminus of the transmembrane IgM, and these code for the hydrophobic sequences. So hydrophobic amino acid residues. So these two M1 and M2, they form this carboxy terminus for the transmembrane IgM. And since we do not require these hydrophobic amino acids at the carboxy terminus of the secreted IgM, so these two are spliced out in the secreted IgM, and therefore the carboxy terminus of the secreted IgM contains hydrophilic amino acid residues. So here we can see that clearly alternative splicing, due to alternative splicing of these two sequences M1 and M2, the molecule, the IgM or any immunoglobulin, they are either formed transmembrane or secreted. So that is how alternative splicing is carried out, and that determines whether the BCR will be transmembrane or it will be secreted as an antibody. Now, coming back to the question once again, after discussing all of these, now let's see which options or which statements are correct about BCR and antibody. So BCR and antibody have identical antigen binding site. This is clearly right because we mentioned already that they are formed from the same gene and they can bind to the same antigen molecule. So this is definitely a correct option. Coming to the next statement, which is BCR and antibody recognize different epitopes. So if they have identical antigen binding site, they will definitely not recognize different epitopes. So if A is correct, B is false. So therefore, B is the not the correct option. Okay, now coming to option C, which is BCR and antibody are encoded by the same gene. This is also something we have discussed. BCR and antibody are coded by the same gene. So this option is also correct. And the last one, that the BCR and antibody are formed by differential splicing. This also we have discussed, that BCR and antibody are formed by alternative splicing or differential splicing. And this is also a correct option. So therefore, the answers to this particular question will be A, C, and D. So that's all. Thank you.